Good evening, everyone. I am Someli Akhtar, speaking from Alamkul Institution, and we are studying about the reproduction in animal chapter, and we have studied about asexual reproduction in plants and natural vegetative propagation. We have studied in our class, and few points from them are left, and then after that we will move forward to artificial process of asexual reproduction in plants. Okay, so whatever is left, first let us continue with that. We have studied about that stolon, suckers, offset, and everything, which are underground stems, which are under modification of roots, which are modification of leaves. They are helpful in vegetative propagation in plants, and it is a mode of asexual reproduction in plants. Now, some of them are using the method of zoospores or conidia, which are basically single-celled plant organisms, and under unfavorable conditions, they use these kind of methods. Okay, so let us go through this. Under unfavorable conditions, some plants reproduce asexually through spore formation. Spores are what? They are minute and thick or thin cell walls रहते हैं, and they are spores are capable of forming new individuals. Okay, so spores are formed either by zoospores or conidia. Zoospores are what? They are motile structures with one or two flagella like this. They are what? They have motility in their structure and motility. They are mostly seen in fungi or some of the algae like Chlamydomonas or Eulothrix. What happens under unfavorable condition in the plant, in a closed cell, zoospores are formed, and when unfavorable condition has gone, then these zoospores have the motility and they will separate from their parent cell and they will develop into a new whole organism. Okay, then by conidia, they are the non-motile exogenous spores. See, they will just germinate over the main plant body, and after germination, this one part will cut off from the whole parent plant and will fall off the ground or fall off the other area, and from there it will grow as a separate entity into a plant. Example is Penicillium or Aspergillus. Okay, now we will see about artificial vegetative propagation, which is done with the help of man. These are what? Man methods propagation. These are the suitable for what? Man create changes in the plant for the development of new plant, and new hybrid and new kind of plants can develop. Okay, so first method is what? Cutting. Cutting is what? This is a small piece. In this, a small piece of any plant organ is used for propagation. See, cutting is what? From this picture only it can be understood that we are cutting a small portion from the parent plant, and from this small portion we are taking this small portion into a new plant body, new soil, new environment, and from this small portion a new plant will be developed. Okay, so this is what? A method of cutting. Cutting creates what? A large number of plants of the same kind, like we can create a clone of that plant. Okay, so example is Bravophyllum, roots, stems, sugarcane, etc. Tamarind, sugarcane, many of the plants are there which we use the cutting method and for the growth of clone plants or for the plant copying. Okay, then layering method is there. Layering method is done what? In this method, a small portion of roots are induced on the stem branches, and when they are grown a little bit more, the stem part is cut and planted in form of a new plant. What happens? Like if this is a plant, so they are having the stem and flower, everything. So in basically layering happens what? A small portion of soil is being attached along with the foil paper or polythene to the stem, some part of the stem, and after some time you will observe that roots start to develop from that area of the stem where the soil has been covered. Okay, and then after the evolution of roots and after the formation of root in that particular stem part, what happens? It has been cut by the man method, and then after cutting they will plant this whole area into a new pot or new area, and then this will develop into a new plant. Okay, one plant is already there, and by the help of layering, by the development of another soil method sticking on the stem, another plant has been developed, and now it has introduced its clone. So this is what happens in layering. Okay, now grafting. In this part, one part of the plant is taken and attached to the other plants as two plants merge to grow into one plant. Okay, in this happens what? This is one plant and this is the other plant. इसमें क्या होगा? One part of the plant is cut as so, and the other plant is cut like this, and after cutting ये दोनों का ही part क्या हो जाएगा? Mix हो जाएगा. Plant A ये है और plant B. Okay, so one part is downside like this, other part will be attached like this. from this is B, this is A. So two separate plant entities will combine together and they will form a new plant. Okay, this is done to increase the number of plants. It is done in rose, plum, and peaches. Okay, so this is done in the grafting method. Now, micropropagation, most famously used method is this one. Cell, tissue, or organs part of the plant is taken and are grown in the laboratory in form of culture. This is called as tissue culture. Initially, it starts with the formation of callus, which further forms many plantlets, each one capable of forming new plant. What happens in this micropropagation? A cell from the plant is been taken and then it will be developed in the laboratory method. ठीक है, cell को grow कराया जाएगा via laboratory method and it will form several new cells. Then from these cells they will form tissue. Tissue will be formed which will contain group of cells like this. And then from this tissue, when they tissue culture type means कि इतने small enough plant create हो जाए so that they can be planted in a soil, under the soil. Then they will release that tissue or that callus culture into the environment for their growth. So this is micropropagation, which is done initially in the laboratory. Then after the growth at particular stage, they will be taken out and will be planted into another pot. Okay. So this is what micropropagation. This is all about the artificial asexual reproduction in plants and animals. Now we will study about the sexual method of reproduction in higher organisms. Okay. So we will just discuss the basics and starts with the sexual reproduction in plant from the next class. So we will discuss the basic. What is asexual mode of reproduction? The mode of reproduction where fusion of male and female gametes either by same individual or by different individual of opposite sex is known as sexual reproduction. Sexual reproduction means what? A and B, two parents are there. Then they will release their gamete like this. Male will release its male gamete, then female will release its female gamete, and then they will both combine, and then fusion will occur. After fusion of gametes, they will form zygote, and then zygote will be able to form a new individual. It will grow and form new individual. So this is how sexual reproduction is done. What are the characteristics? Let us see one by one. As fusion of male and female gametes are involved, the offspring produced are neither identical to their parents nor among themselves. This is the main reason of the sexual reproduction. They create genetic variability among themselves also and from their parents also. It is an elaborate and complex and slow process as compared to asexual mode of reproduction. Why it is an elaborate and complex? Because it will take time for the formation of gametes, for the fusion and for the growth. So that is why it is complex as well as elaborate time-taking process. It helps in genetic recombination. Because why it is helping genetic recombination? क्योंकि ये difference create कर रहा है. None of the parents will be identical to their daughters or offspring, and none of the offspring will be similar to their siblings. Then it plays important role in evolution. Why it is playing important role? Because it is creating genetic variation, and through genetic variation, evolution is occurring. This kind of reproduction is exhibited by multicellular organism. All the flowering plants and most of the animals. Means what kind of animals and plants are showing? Those who are multicellular and higher in their hierarchy, they will be able to reproduce through sexual methods. Then the period during which an organism grows to attain maturity is called as juvenile phase and vegetative phase. Juvenile phase in animals and vegetative phase in plants. Means up till the period, there are certain period limit कि इस period तक attain करने के बाद ही they will be able to reproduce sexually, and before that period it is called juvenile phase in animals and vegetative phase in plant. Because these phase they will not be able to reproduce through sexual method because they have their gametes have not matured, they don't have a certain level of energy, they don't have that kind of hormones. So up to this phase they will not produce through sexual method, but after this phase they will be able to after attaining the maturity, they will be able to reproduce sexually. When the organism starts reproducing sexually, it is called as reproductive phase. जब sexual phase आएगा तो उसको बोलते हैं reproductive phase. When the reproductive phase ends, it is marked by the old age phase, that is senescence phase. Each of these phases are regulated by hormones. Okay, start कहाँ से हो रहा है? Start होता है juvenile phase से. Juvenile phase से start होता है. Then यहाँ पे sexual reproduction नहीं हो रहा है. Then आता है maturity phase और reproductive phase, and then after a certain point of view maturity जब old हो जाती है तब आता है senescent phase. So these three phases come under the whole lifespan of an organism, and reproductive phase is the only phase where sexual reproduction occurs. Reproduction Okay. So that's all for today's class. From next class, we will discuss about sexual reproduction in plants and animals. Okay. Bye.